At the Leibniz-Institut of Plant Genetics and Crop Plant Research at Gattersleben, Germany, a highly innovative plant cultivation facility is currently under construction, the Plant Cultivation Hall. It is specifically designed for high-throughput phenotyping, with plants growing under conditions that are typical to the field, but which can be imposed in a highly reproducible manner. Unlike in field trials, environmental conditions such as light input, air temperature, CO2 concentration, and air humidity, as well as water and nutrient supplies, can be precisely set and regulated.
Well, our main goal is at IPK to link genetic variation of the genome of the plants with the expression of traits of their features, like yield production, or also the tolerance and resistance against environmental conditions and extremes that more and more occur in nature. And we will be able to simulate conditions that happen outside, but we will be able to reproduce them precisely and modulate them according to our experimental needs. The experiments that may be carried out or will be carried out will test plants of a certain genotype in different environments, like drought stress conditions, for instance, or variation in temperature that may occur even in future due to the climate change. And on the other hand, to see whether different genotypes, different genetic constitutions of plants modulate or influence the expression of these performance traits in different defined environmental conditions.
The Plant Cultivation Hall will comprise 2 different phenotyping systems in an area of almost 500 square meters. One is a container-based system for plant stands, the other a so-called rhizotron-system for individual plants. The rhizotron system is being developed and implemented in collaboration with the Forschungszentrum Jülich. It will support the simultaneous cultivation of up to 360 plants and will enable very detailed studies of the root architecture. Imaging towers equipped with specialized lighting and camera systems automatically move along the rows of rhizotrons. They successively take out individual rhizotron boxes to image the plants. The images are digitally evaluated, providing information on the characteristics of the root architecture, as well as on root and shoot growth. The other area of the hall houses the container system for the phenotyping of stands of plants of vegetation. Various camera and sensor systems are mounted on a robotic crane that can be precisely positioned. These plant imaging systems use different wavelengths and scan them in 3 dimensions. The soil in the containers resembles the natural soil structure in the field. Its temperature can be precisely controlled at different depths. The temperature and soil moisture are monitored by numerous sensors. Built-in imaging tubes allow the recording of root growth processes.
So among the main tasks of IPK is the valorization, the analysis and characterization of our vast diversity that is kept in the GenBank, in the seed stock collections of our crop plants. And so this cultivation hall with the phenotyping platforms that we will establish here will allow us way better to characterize and describe the properties of these resources and thus to help their utilization in future. Furthermore, we are deeply interested in analyzing the mechanisms that lead to the performance traits of plants and thus help to improve agricultural production in an environmentally friendly fashion in terms of higher productivity in agriculture towards the production of sufficient food and feed, as well as renewable resources and energy for future generations, actually.
The IPK's Plant Cultivation Hall is a worldwide unique infrastructure. It sets new standards in plant research while providing an excellent basis for national and international collaboration and joint research projects.